Alright, let's start off with a Unit 5 review. I have tried recording this entirely too many times, so we're going to try it in one go. If you hear any weird noises, my windows are open, so that is what that is, and I have had motorcyclists running around here. So, I'm going to skip this and reference back to it after the next slide, because this is really basic biology. But we'll start with the P generation. The P in P generation stands for parental. It is the one you originally start with, which is this one right here, which is your heterozygote and your homozygous dominant individuals. It's just whatever they originally give you to cross. The F1 generation is going to be what comes out of this cross. So in this case, it'd be whatever individuals are coming here, their offspring. F2 would be the results if I crossed anything from the F1 generation, and then so on and so forth. So, dominant and recessive alleles, this is only for simple Mendelian genetics. This is the basic stuff you usually learn in your freshman year of high school or whenever you first take biology. So to start with, an allele is a form of a gene. It's just basically what version you have. It's just a section of DNA. A dominant allele will always be a capital letter, and for simplicity's sake, I'm just going to use G for this one for green and yellow seeds, like Mendel's classic experiment. But a dominant allele will always be shown as a capital letter. It will always be expressed physically when an organism has it. So if I have like a capital G, no matter what, if I'm a plant, because that's what I decided to do that day, I will always have green seeds because I have a dominant allele. If someone's very domineering, they tend to be very controlling, kind of in your face-ish, depending. We've all known that person. But recessive alleles, on the other hand, will be expressed as a lowercase letter. So in this case, we're going to have little g here, or here. These are only expressed if they are not paired with a dominant allele or a capital letter. So as long as I have one capital letter, I will always show the dominant trait. But if I have two lowercase letters, then I will show the recessive trait. To recede means to like run away. So it runs away or hides from the dominant allele. So genotypes, just like geno implies, genotypes are the genes or any possible allele. So there are three possibilities you can have. There's homozygous dominant, which has two capital letters. Remember, homo means same. So I have two of the same letter, but they're both dominant, they're both capital. I have heterozygous, which is one capital letter and one lowercase. Hetero meaning different, so I have two different letters right there. Then I have homozygous recessive, which is two lowercase letters. Again, homo meaning same, so I have two of the same letter, but because it's recessive, it's two lowercase letters. Homozygous individuals can also be called true breeding. You may see that phrasing pop up on the exam or tests or something like that. It just means they're completely homogeneous, they're completely the same. Heterozygous individuals can also be called hybrids, but that can be a bit tricky because hybrids mean something else when it comes to like species and stuff like that, which we'll learn later in the year. When we write genotypic frequencies, we write it homozygous dominant to heterozygous to homozygous recessive. So if you did the Punnett square for these two, you would get two individuals that are homozygous dominant, you would get two that are heterozygous, and none that are homozygous recessive. Because, plot twist, if you have one individual who's homozygous dominant, it is impossible, obviously, for you to have two little b's at any point in a monohybrid cross. It's impossible if you have two little b's, so everything will have at least one capital letter. That being said, phenotypes are the physical characteristics. It's what you actually see. The dominant traits will always show when there's at least one capital letter. So this would include homozygous dominant, because I have at least one capital letter. In fact, I have two. And heterozygous, which is one capital letter, one lowercase, but I still have that one capital. Recessive traits, on the other hand, will only show if there is no dominant allele, like I said earlier. The only genotype that you can show is recessive, that can show recessive, is homozygous recessive, which is, in this case, would be little g, little g. So if I go back to this individual, if you did the Punnett square out for this, no individuals can have two lowercase letters. So when I write this phenotypically, my ratio, it's dominant to recessive. So because they all have at least one capital letter, every single one will have whatever this trait is. I didn't make one up. But because none have two lowercase letters, none will display recessive. So that's how, I use the simplistic example, which technically is not true, but human eye color. If someone has blue or green eyes, which are the recessive ones, then you can know what their genotype is. But as we'll see later, this is not actually how that works. It's just an easy example. Alright, so the type of inheritance we just talked about is simple Mendelian genetics, or just complete dominance. It's simple inheritance. The dominant will always show over the recessive, no matter what. But then all the really fun, scary stuff is called non-Mendelian genetics. So that's where things get complicated, because very, very few things actually follow this complete dominance pattern. A lot of things are much more complex than just that. So codominance is when there is no recessive allele. Co means together, dominance for the dominant allele, so they're both dominant. They will both be expressed at the same time, but they don't blend together. So for example, for 3 o'clock flowers, or whatever type of flowers those are, red and white flowers will present as like striped. That's how you can get certain patterns like on cows and stuff like that, because it's codominance. Incomplete dominance is when neither allele is completely expressed. It's kind of like when you put two passive people in one room, they can't really come to a really good decision, so they just kind of blend together. So neither is truly dominant, but we still write it as a dominant. So for red and white flowers, they would present as pink, because red and white is pink. There is another type of codominance that pops up rather frequently, which is blood types. Blood types, and I forgot to include it in this presentation, they are codominant, but they also have multiple alleles. Multiple alleles is when you have more than just the two basic dominant recessive type alleles. So for codominant, I have one that codes for type A blood, I have one that codes for type B blood, and those are both codominant. However, I also have a little I, which is technically recessive, so I have 3 alleles, hence the name multiple alleles. So things can combine all these traits together. It's a really great cluster. So if I have red and white flowers, if it was incomplete, neither of them are being fully expressed, they can't really make up their minds, so they just blend together. Red and white is pink. For codominance, they both are duking it out and trying to figure out which one is actually going to be expressed. So they'll come out striped or spotted or something along those lines. Epistasis. Epistasis involves multiple genes, but it should not be confused with multiple alleles. It just means that there are many genes that are working together to an extent. So for example, coat color in animals is one of the most common examples. So I can say I have something that colors the coats, whether it's black and yellow. Um, I want to see if a mouse will have a black or yellow coat. However, there is a gene that shuts it on or off. So let's say if I have a capital A, that would make my organism albino. So first I have to see if it would have a color at all, and then I can go on and determine this. So if I had at least one capital A for this example, my organism would be albino. Doesn't matter. This no longer matters at that point. But if I have something with two little a's, then I have to go to the next step and figure out what coat color it'll be. So it's basically this switches one on or off, and then if it's on or off, depending on whatever you're looking at, you go on to the next step and figure out coat color or blood type. Not blood type, that's a bad example. I don't know, something along those lines. So we have polygenic inheritance. This is not to be confused with multiple alleles. Multiple alleles is just when I have more than two, dominant recessive. I could have two recessive, two dominant, three dominant, whatever. This one, on the other hand, is a type of inheritance when basically many different things chain together and can control one trait. So it depends if these are on the same chromosome or things like that. So this is how we have things like human eye color. They're all varying shades. This is aside from heterochromia, which is different eye color. This is how we all are different shades of skin colors. We have different heights. For example, my biological father and my half-siblings, for the most part, are on the taller side, but my mom and I are on the shorter side. My genes just did not stack up correctly. Then we have sex-linked genes. Something is sex-linked, it obviously is on the X or Y chromosome. So a Y-linked gene can only affect biological males, because as a biological female, I do not have a Y chromosome. I'm incapable of getting anything from that. Usually the Y chromosome, for the most part, only codes for sexual characteristics, which is just puberty, basically. The X chromosome, on the other hand, codes for a lot more. So you'll see, actually, if you look at it in a karyotype, which, if you guys remember, is a map of DNA, like the glowy map or whatever, where you see all the chromosomes lined up. If you see it, the X chromosome is actually bigger than the Y chromosome because it codes for more than just the puberty characteristics, for simplicity's sake. But X-linked genes can affect both males and females, because remember, you cannot survive without an X chromosome. So these genes affect absolutely everyone, which will make sense when we talk more about pedigrees. However, because biological females have two X chromosomes, in order to prevent too much gene expression or too much of the same genes, one of the X chromosomes will actually shut down or inactivate, which is called a Barr body. What happens is it almost kind of shrivels up and gets kind of weird and doesn't really express because there's no reason for it to. It's unnecessary. This is how you get things in calico and tortoiseshell cats. Some of their color is controlled on the X chromosome, so it'll shut down, and then you get that really nice coloring on them. So then we have the laws of heredity. The law of segregation says that two of the alleles for the same trait will separate during, or gametes will separate during creation, end up in different sex cells. Basically saying that when you're creating a gamete, you're putting one version of an allele, let's say the version for green, or I'm a homozygous, or a heterozygote for green and yellow seeds. My allele for my dominant green seeds will go to one gamete, and then my allele for yellow seeds will go to another. So it just has to split. The law of independent assortment says genes from one trait will separate independently from another. So just because I have brown hair doesn't mean that it'll dictate if I have brown eyes or not. They're completely independent. They do not affect each other. Because remember, this goes back to simple Mendelian genetics, where we didn't know much about epistasis and polygenic inheritance and all that good jazz. So we have asexual versus sexual reproduction, or mitosis versus meiosis. Oh, okay, I guess I can. So asexual is one parent that creates two genetically unique clones. There's no gametes, nothing like that going on. I just bloop off a little piece of myself. The only way that I can get genetic variation or I can change my gene pool is through mutation. I don't have an option because everything's exactly the same. So this is mitosis. This happens also in somatic cells, which is everything but your sperm or egg cells, which is later in here somewhere. Sexual reproduction has two parents. I have a sperm and egg cell is the creation of sperm or egg cells. The offspring are unique combinations of genes from parents due to crossing over. So they're genetically varied from parents and siblings. You can be very similar to your parents, but you are not exactly the same as them. Or else that'd be really weird. All right, pedigrees. Pedigrees are family trees that can show inheritance or how things are passed down. Horizontal lines show mate or parents. I say mates. I know it sounds weird, but that's just because technically you don't have to be married or anything like that to have kids. And then we can also use other things that don't get married, like plants. I mean, they could. Vertical lines show parents to offspring or parents to children. Generations are measured in Roman numerals. You'll usually see them off to the side, whether it be left or right or whatever. And then individuals are listed from left to right in terms of oldest to youngest. It may reset depending on who made the pedigree or what pedigree you're looking at. Some numbers may reset between generations. Like, let's say I have two parents, it'd be 1, 2. Then when I go to their kids, it'd be 1, 2, 3, 4. Or sometimes it will just go ahead and not reset, and so the parents would be 1 and 2, and then the kids would pick up at 3, 4, 5, 6, and so on and so forth. But it really depends, I think, on who made it. For the age arrangement, it is only the biological people of who is actually married, or who is actually in the family, sorry. So it does not include people who have married in or mated into the population. So I could have someone on the far right, who would be the youngest, married to someone who's much, much older than them and the rest of their family, but because they are not a part of the biological family, they don't count. So circles represent biological females, squares represent biological males. If something is filled in, that means the individual is quote-unquote afflicted, or it means it has the trait being tracked. It's not always about diseases. That's just the easiest example. I could be tracking the gene for bad breath. I don't know. And I could track that down. So it's not, that was also not a good example. But I could track that down through a family. It's not a disease. It's just, yeah. If a circle or square is not filled in, that means they do not have the trait being tracked. So that means they're unaffected. Carriers can be represented as like slashes, not through them, but like through the middle, like half is colored in, half is not. Sometimes I've seen it where they don't color them in, and so you get to play a fun guessing game if they are affected or not. Sometimes I've seen them completely filled in. So it can get a bit wonky, but for the most part, if it has a slash, if it's half colored in, half not, that means they're a carrier. They themselves do not pass on, or they do not have the trait, but they can pass it on to the next generation, which is important in a little while. So, when we're reading pedigrees, autosomal dominant disorders affect males and females equally because an autosome is a non-sex chromosome. Does not control your biological sex, so it doesn't matter if you're biological male or female, you're just as likely equal to get this. If something's a dominant trait, you usually see it in every generation or extremely frequently. If your parent has the trait, there is a high chance that you are going to have it, but you may not necessarily have it. However, this means if you do have it, that means at least one of your parents has to have it. So that's like cleft chins is a good example of a dominant trait that we could track through. If you have a cleft chin, at least one of your parents has to have a cleft chin. Please don't go check. I don't want anyone to find out they're adopted through me. An autosomal recessive disorder affects males and females equally, because remember, it's an autosome or a non-sex chromosome, but it can quote-unquote skip generations. It does not necessarily skip, it just hides, because if we remember, recessive alleles will always hide to the dominant. So that means if it does skip a generation, whoever had that allele, and it reappears, whoever had the allele had a dominant copy that masked this version. Recessive disorders will have carriers that are heterozygous. Like I said over here, carriers do not effectively they do not present the trait, but they can pass it on. So it's possible that a parent doesn't have the disorder, but if they have a dominant copy of the gene, but they have the ability to pass that on. So an example is like cystic fibrosis. You may not have cystic fibrosis, but you do have the ability to pass that gene down. Sex-linked disorders, as the name implies, it deals with your sex chromosomes or your X and Y chromosomes. In sex-linked disorders, one biological sex is affected more than the other. So in X-linked dominant disorders, more females will be affected than males, because as a biological female, you would have two copies of the X chromosome, so you have double the chance of getting whatever the disorder is. If you have an affected father, if a dad has it, then his daughters will all have to have it, because remember, the father is the one that determines biological sex. He can give either an X chromosome or a Y chromosome. But he gives me an X chromosome, I'm a biological female, but if he has the disorder, that means I have to get the defective copy. If a son has the disorder, that means he has to have an affected mother, since they get the Y chromosome from dad. X-linked recessive disorders usually affect males more than females. A female must have two copies of the X chromosome to have the disorder. So if I get one copy from mom, or from dad, that's defective, I have to get my other X chromosome from mom, and so the defective one, or the one that has the disorder, will just shut down and my other one takes over. I need either my mom to be a carrier or also have the disorder, and then I would need my dad to have the disorder in order for me to get it, because I have a built-in biological backup plan. However, males do not have a biological backup plan, since they have only one X chromosome. So, oh crap. I actually mixed this up. Please ignore this one. My bad. But if a mom has the disorder, her son will have the disorder. But mom can also be a carrier and give it to the son who has no backup plan, and will have the disease. So this is things like hemophilia, color blindness. That's why statistically men are more likely to be red-green colorblind as opposed to women. Is there a way I can scratch this off? I don't know how to use this computer. Okay, ignore this. Ignore this. I was supposed to go up here. Oh wait, no. Sorry. Okay, I know what I meant. Don't ignore this. I have lied to you. But like I said, if one's dominant, it'll turn to a bar, or the affected one will turn to a bar body. So all daughters of affected fathers will become carriers. So if my mom doesn't have it, but dad does, that automatically means, so like color blindness, I won't have color blindness because I have one healthy X copy. But if dad has it, that means I have to get it from him because he determines my biological sex. So that means it'll be passed down. One that I don't think I included, I did not, is a Y-linked disorder. A Y-linked disorder can only affect biological males because, as I said in that earlier slide, I don't have a possibility of getting that as a biological female. I'm XX, whereas a biological male is XY. Then we have mitochondrial inheritance. DNA from the mitochondria and chloroplast are inherited from the mother, specifically through the egg cell or the ovum, ova, however they phrase it very differently each time. So you get that from your mom, because your dad has sperm cells, not ovum cells. So these traits come from only the mom, and all offspring run the risk of being affected. So if your mom has a mitochondrial disease or disorder, there is a 100% chance you will also get that because she is the person who provided the egg for you to exist. So daughters will always pass that down to their offspring, but the fathers will not. So as I was mentioning different types of chromosomes, we have an autosomal chromosome or an autosome. This codes for anything besides biological sex. As humans, we have 22 pairs. They code for things like hair color, eye color, metabolism, things like that. Then I have a sex chromosome, as the name implies, codes for your biological sex. In humans, we have one set. It is possible to be born without a Y, but you cannot be born without an X. A biological female is XX, biological male is XY. And then, very similarly but different, these both mean very similar things, but these are chromosomes, these are cells specifically. A cell cannot be a chromosome, but cells do have chromosomes. So a somatic cell or body cell is anything that's not specifically an egg or sperm cell. Most of your cells are somatic cells, with things like your eyes, heart, liver, etc. Gamete cells or gametic cells are also known as sex cells. These are exclusively sperm or egg. These are the ones that undergo meiosis. These are the ones that undergo mitosis, or asexual reproduction. Helps you heal and grow and get larger. These are used exclusively for sexual reproduction. And then haploid versus diploid. A diploid is basically a normal cell. It has what's called 2N, or 2, whatever your number of chromosomes are, which is a complete set. These ones, when you look at them in the cell, they have an X. That's that X shape. These are any cells that undergo mitosis or asexual reproduction, aka somatic cells. A haploid cell, if you really don't think about it, haploid kind of sounds like half, but it has N amount of chromosomes. This is only in sex cells. These undergo meiosis. So this is your sperm and egg cells. This is every other cell in your body. So I'm not going to go through the exact every single step of meiosis 1 and 2 because it's very, very similar to mitosis. You guys know those steps, so I figured I'd just highlight the key events of meiosis 1. So there's 3 main events in meiosis 1. The biggest one is prophase 1. This is where crossing over happens and synapsis actually occurs. So this is where your homologous chromosomes, your tetrads, attach to each other and exchange genetic information. This can happen at several points. It doesn't have to be the classic where it's just like one singular arm that is exchanged like when I hold hands with the skeleton Timmy. It can happen at many points. And then this increases genetic variation. This is how we are all similar to our parents, but not exactly the same. It's extremely, extremely important for our population to survive and be genetically unique. That's how we've lasted for so long. Metaphase 1 is where the tetrads of those homologous pairs line up at the metaphase plate. Remember, meta means middle, or M and M. And they're just sitting there. So at this stage, you'll see two homologous pairs, or basically like 4 sets of chromosomes, sitting right next to each other, lined up at the plate. Sorry if you hear stomping. Oh. Sorry if you hear stomping. My husband is playing the guitar, which is apparently the loudest thing on the planet. So I'm hoping he hasn't switched to drums. Then we have anaphase 1, which is where the homologous chromosomes that have lined up at the metaphase plate then separate and go to the edges where they go to meiosis 2, which quintessentially looks like mitosis. The big thing about meiosis 2 is that the DNA does not duplicate again during interphase, because if it duplicated, I'd be back to step 1 and have to do all this over again. Things would get real messy real fast. Oh, Jesus. Oh, yeah, beautiful picture. So as you guys can see here, I have my homologous chromosomes holding hands. They're having a grand old time. Then here they're lined up at the plate. So there's technically 2 chromosomes for red, 2 chromosomes for blue on each of these. That's what I say when I mean there's 4 of them in one of these little steps, because I have 2. It's just hard to tell with this wonderful blurry picture. Then they separate during anaphase 1. The tetrads go to each side. They look all so jazzed about it. As you guys can see, this blue has a little bit of red mixed with it, and that came from the crossing over. Like I said, it's not always just that one singular point. Usually they attach at multiple stages. And then I move on to telophase 1, where they start to split in cytokinesis, and I move on to meiosis 2. So chi-squared is statistical analysis. I use it to see differences in data, to see if what I'm looking at has something affecting it, or if it's just random chance. Because remember, we don't live in a perfect world. Every time I flip a coin, I'm not always going to get an even 50-50. I could get, some more than the other. So chi-square values are compared against what's called the critical, or p-value, to see if something is causing change or not. I need to see if, I'm running an experiment to see if my new painkiller works for headaches. I need to see if my painkiller is doing something, or if it's just all in my head. Pun not intended. So the null hypothesis, or H0, states that any differences seen is due between the actual data and predicted data is due to random chance. So let's say I go back to the coin, I flip a coin 53 times, heads up, and 47 tails. Well, that's not too far off. I could say, yeah, that's just random chance. It's just, how the coin ended up flipping. However, if I was using a weighted coin, you'd see the results were really, really off. And so I would have to run an experiment to see, hey, is this normal or not? So for my headache, or my headache pill example, I would run an experiment and then run analysis to see, maybe 30 people, it helped their headaches, but 5 more people were like, this didn't help my headache at all, it's worse almost. So I'd be like, okay, did my headache pill actually work, or was everyone convincing themselves that it worked, or are they convincing themselves it didn't work, or So chi-squared, I use the x2. I didn't really feel like putting the fancy x in, and the little square up here, that's just a lot of effort. But that's the value that you actually calculate. That you will be given the formula on the exam, but you won't be given the table. So it'll give you the sum of that really scary looking formula. but it'll give you your observed and hopefully your expected values. That's what I've seen lately. I haven't seen them ask you guys to calculate that out, but I'm just telling you so you know. Then your critical value or your p-value is that chart that is given to you. You will be given the chart. You're not expected to memorize it. You calculate it at the first or at the 0.05 level of certainty. So you'll see like p-value, p.05. Look at that column. That's the one you need to look for. In fact, I think they stopped giving you the 0.01, the 99% confidence. But what that means is that I am 95% confident that what I'm seeing is random chance. Any data that falls in this category, in this chart, I'm 95% sure that it's random. Nothing outside is having an effect on it. So when I do compare them, if my chi-squared value is larger than my critical value, then you reject your null hypothesis. Because the null hypothesis says that everything's due to random chance, that means that if I reject it, I have crossed a critical threshold or a critical line, and I'm like, hey, something is causing this difference. I'm pretty positive because I've gone way too far outside the region of where I feel safe and comfortable of my 95% confidence to say that something is not causing this. So I think something is happening. This means it's statistically significant, which I should have included, but we'll do chi-square again. But that means it's statistically significant. That means I 1,000,000% am doing something, or something is causing a change. So for my headache medication, I run the test and I'm like, oh, I fell outside of my chi-squared values. So, I know my medicine's working. I know my medicine's making a difference. You can't tell me otherwise. Whoop-de-doo, and then I can charge you $100 for it. Or I could say the weighted coin. So if I used a weighted coin and I'm flipping like 70 heads versus 30 heads, it's like, whoa, wait. In theory, I should be getting a perfect 50-50. Something's going on here. Something's a bit fishy. If my chi-squared is less than my critical value, then I fail to reject or I accept the null hypothesis. This means, yeah, sure, there's some differences, but I can't confidently say that there is an outside force working on this. So if, for the coin thing, you could do the 53 versus 47. It's like, yeah, that wasn't a perfect 50-50, but it still, fell within a reasonable range. And oh my god, we did it. Yay! Alright, thanks for hanging in there with me. Sorry my throat's a bit dry. This has been a wonderful time. Alrighty, goodbye.